train their bodies into our simplistic renditions of the quantum weirdness that is molecular structure. We will loosely follow the path of information transfer from DNA to proteins that exists in all living creatures today. We hope you will leave with a more visceral understanding of these molecules that play such an integral part in our lives. And yes, I too have a lab coat. Phosphate groups are what holds one base to the next in a single DNA or RNA chain. The oxygen atoms on the top and bottom form a phosphodiester bond with the sugars above and below. In addition, the phosphate group is responsible for the net negative charge on a DNA or RNA chain. Deoxyribose is the sugar molecule that forms the second part of the DNA backbone, and it puts the D in DNA. It has one more oxygen than ribose, the sugar used in the RNA backbone. The sugar is in a single strand of DNA, all share the same orientation and used to distinguish the strands. Here, Ellen's outstretched arm is the 5' OH, while Carrie's ankle is the 3' OH. In a longer strand, these two oxygens will form the attachment points for the phosphate. The bases in a DNA or RNA store information in the form of the genetic code. The four bases, adenine, guanine, cytosine, and thymine, or uracil in RNA, are arranged in patterns that encode all of the proteins in your cells. This is cytosine, which bonds with guanine. Once these are combined, they form a nucleotide. A nucleotide is the functional unit of DNA, comprised of a nitrogenous base, a sugar, and a phosphate group. If we can have a couple volunteers from the audience, I think we can... Okay, sit down. Talk to Carrie after the show. So what can you make with only cytosine, you ask? Proline. The codon or base triplicate encoding an amino acid for proline is CCC. This unique amino acid is an essential component of many important proteins. Its fixed ring structure causes a kink in the peptide chain of a protein. If you repeat a lot of prolines in a row, you form a polyproline helix, the principal component of collagen. Glutamine is another amino acid, encoded this time by CAA. It serves many functions, from protein synthesis to serving as a backup energy source for muscles. In human blood, glutamine is the most abundant free amino acid. Glutamine is not to be confused with glutamate, an important neurotransmitter with a similar structure. Cysteine is an amino acid that serves an important structural function in protein. Cysteine is one of only two amino acids containing sulfur. The other is methionine. The sulfur atoms in two neighboring cysteine residues can form a disulfide bond. These bonds are often necessary for folding a protein into the correct shape. Now we leave behind the central dogma of biology and move on to a pair of small molecules relevant to our gathering tonight. When glucose is metabolized in the absence of oxygen, it produces two molecules of pyruvate through glycolysis. Pyruvate can then be converted in animals to lactic acid through anaerobic respiration. Yeah, they're also demoing lactic acid, I think. Plants, however. In plants, anaerobic respiration metabolizes pyruvate into this molecule. If you look closely into your glass, you may see it swimming there. But if you've had too much, you may have lost the ability to focus that sharply. Circus, and a wonderful audience. Thank you.
Participation, and then we'll follow up with mathematicians behind the music. So.